I'm happy to be here, even in short notice, to discuss about FLIM and Luminosa, both things that they are very close to me. So I will try to make it fast. Luminosa was launched close to 2 years ago. It's a single photon counting confocal microscope. We put single photon counting even in the description of the microscope to differentiate from the other great confocals that they are there, because Luminosa, if you want to differentiate with different microscope, it has at its core of its design, I have to say, all the time resolve techniques. So we're talking about single molecule FRET, single molecule imaging, FCS, and then FLIM, FRET, even anisotropy imaging. These are methodologies that maybe for other confocals, they are still in the region of being upgrades. For Luminosa, they lie at the heart of its design. So it's designed with a focus on biophysics, but as biophysics going more and more into the direction of more complex environment like the cells, one can say that, and we expect that Luminosa will become more and more relevant for the cell biologists, because they are also, for many assays, one wants to start from the molecular scale and go to the cellular scale, and it would be nice if one can do it with the same instrument. A nice example of this approach is, for example, the study of liquid phase separation in intrinsically disordered proteins. There one can start by studying each individual molecule in vitro by single molecule FRET or FCS. But then, of course, one can use FLIM when one wants to study this assay in the cellular environment. But in this talk, I will not focus about the other parts of Luminosa, like single molecule FRET and so on, and FCS. I will focus on FLIM, and I will try to see, to give you an idea how we push the boundaries of confocal FLIM in different directions. So first, we're talking about being more quantitative and more reproducible than ever before. And this is not something that is done only on the software side. So this is a combination of hardware and software, as you will see. So we have the best in class dead time electronics. So we have zero dead time in the collection efficiency. We offer the possibility to either work with hybrid PMTs or with SPADs, which are more sensitive in the NIR. The optical design is made for high sensitivity, so we have no fiber coupling and so on in the detection pathway. And on the same side, simultaneously, we have developed some GPU-accelerated algorithm and some context-based workflow that they make the acquisition and the analysis more reliable. So I'm giving you this screen here just to tell you that when one starts, Luminosa gets this screen, and you don't have to go immediately in the optical path. And if you select this context-based workflow, then you give some information to the instrument about what you want to do, and then the instrument can guide you if this is necessary. Still, the expert has complete freedom, but you have this default context-based workflows that they act as a safe haven for the inexperienced or maybe for the newcomer. So, for example, one can select the FLIM application, and then after selecting the fluorophore, then the lasers that they are needed and the repetition rate is selected automatically by the software itself. What I want to stress here is not only the detection. What I want to stress here is, if you see down here the analysis channels, you see that we work in the pulsed interleaved excitation. So you have the blue and the red laser alternating very fast. So we can say that we are truly simultaneously acquiring our data. The Luminosa has an auto alignment function that takes 2 clicks and less than 1 minute to be applied. And one can say, oh, why you need an auto alignment? And the difference is actually the difference between guaranteed and optimal performance. With Luminosa, we want to make to be sure that we're always at the optimal performance. And it could be that if you don't do the auto alignment, maybe you are a little bit off. But if you do it, since it takes less than 1 minute and doesn't require any sample, actually you can do it with the sample on stage. You can be sure that you have you work always at optimal conditions. And from the facility perspective, if you do the auto alignment every time, you can avoid long-term drift. So here you can see that we have measured and we have tested and we have benchmarked this auto alignment procedure with FCS for a period of months, and we can even on purpose included a winter break where we switched on the heating, but still we could always recover the optimal performance of the system. Another point that that increases the reproducibility is that we have a special algorithm for measuring the power, and then we are, I have to say, brave enough to give the power in microwatt. So there is a calibration procedure that if you do it again every day, it takes 10 seconds to account for day-to-day variability of your laser driver, and then you get the readout of your excitation power in microwatt. So we can say that we have solved the internal problem, what is 10% of my power? And this is done without the need of a power meter and so on and so forth. I mean, when we go to FLIM, so this is a typical example. Let me try to start this video. If it starts that as I'm acquiring a FLIM image, I have simultaneously all the necessary online views for judging the quality of the system. So this is the intensity image, then I can switch on the lifetime display, and I can get the lifetime distribution and so on. And then I can judge the quality of the measurement as it proceeds. So let me move. Then when the measurement is over, actually, we have simplified the analysis. And this, we have done it a little bit with the brute force of the GPU accelerated algorithm. So if the analysis is finished, just by double clicking the measurement, after a few seconds, you get the results. For example, this is an example of a segmentation where you have three different approaches. So you can see, I already give you here the results. You see the nucleus, you see the mitochondria here labeled with it's a TOM20 protein, and here is the tubulin fibers. And what I want also to say and stay a bit here is that here, and this is again the difference in PicoQuant, you see the multi-exponential decay and the phasor plot simultaneously. And even a third way of analyzing the data is the pattern matching approach that we have implemented. So you can have actually with a click of a button very fast on your screen the results of the phasor plot, the results of the multi-exponential decay fit, and the results of the pattern matching. And this is let's say my only comment to the amazing talk from Leonel, with whom we've been discussing rather often the last couple of years, is that one doesn't have to decide beforehand. One doesn't have to say I'm a phasor plot guy or I'm a multi-exponential fitting person. One doesn't have to do this. You have what we think that we have accomplished in PicoQuant by having an easy way to move around from the multi-exponential fit to the phasor plot is that we offer an holistic view of the FLIM data, and by this we increase the specificity of the results. Also, what we will see in a moment, one can zoom in, can see that I can draw a population here in my phasor plot, and then I can analyze the multi-exponential fit of this population. Another point is that you could see that in different assays, for example, phasor plot work better if you have a very complicated decay. It could be, nevertheless, it is true that also in other cases, the multi-exponential fit offers more advantages for the cases so that you want to quantitatively determine FRET with multi-exponential decays and so on and so forth. If we move around, I just want to again summarize. We have 4 different analysis routines that they run in parallel. You have a very fast lifetime contrast just by the arrival time of the photons. You have the phasor plot. You have the lifetime histogram. You have the multi-exponential decays that really just by clicking the software fits with 1, 2, 3, 4, 5 exponentials. Then it performs some statistical tests and tells the user, no, you have to use 3 exponentials. So the inexperienced user doesn't have to lose time and think, shall I use 1 exponential, 2 or 3 in this case? The software itself makes a suggestion, and this simplifies a lot the whole procedure. Because as Leonard also said, fitting can be an uncertainty factor for, especially for inexperienced users. And of course, we have something that is patented and used mostly from PicoQuant, is this multi-species pattern matching approach. Is that also this it's a quite fast approach and works very well for species separation and multiplexing. Now, with Luminosa in the software, we try to offer some more advanced options for NovaFLIM. And what I want to say is, if you imagine, if you are familiar with cytometry, where they use scatter plots, or with single molecule thread community, where they use scatter plots, in reality, one can produce scatter plots from the images where you have pixel populations. So what you see here on the right top corner is a 2D histogram of the fast lifetime for the x-axis, and on the intensity on the y-axis. And you can see here, you can draw a ROI there in this histogram, which is quantitative, it's numbers. And then you can see how this corresponds to your image. It's the same reciprocity principle that Leonel mentioned in his talk, and not only that, but you can see also the phasor. So, for example, in this case, this is the row in this 2D histogram for the mitochondria. You see here the phasor plot population, and this comes also to the last question from Alesh. Then, if you select another population, this is the tumbling, and now you see a different population in the phasor plot histogram. So this phasor plot that you see here comes only from this ROI. Or now I can select a third population. This is the background. It's mostly in the nucleus because there was a little bit of DAPI staining, and you see a different contribution in the phasor plot. And of course, you can export these masks. You can import new masks because, as Alice said, we see that in more advanced application, the possibility of handling different ROIs. Maybe you want to export the image, do your AI algorithm, identify your ROIs, and import ROIs in the software. All these capabilities are now possible with the NovaFLIM software. What I want to say is that, of course, we have a long-term commitment for open data. Open data formats, our formats are completely open, and also enhanced metadata. So because we want to keep on being compatible with the open source initiative, not only the one for like PhaserPi, which I find amazing, but also other open source activities in other areas like FCS, single molecule thread, and so on. So we have a convenient export of all the metadata to XML format. And when I'm saying all the metadata, I'm talking about the type of the detector, the type of the repetition rate, the laser power, the objective, all the dichroics that they were used. They can be exported in the XML format, and there is also a selection of metadata even available in the Homero TIFF exports. So and also a unique identifier. So no matter how you name your Homero TIFF, you can always go back to the raw data, which I think that this also enhances a lot the reproducibility and goes along the directions of the Quare PLIM initiative. As a teaser here is that because we have been in many discussions the last 6 or 12 months regarding data formats and so on, and we heard many opinions about how we can become better and faster. Very soon we will have an additional data format. We will not replace our existing data format, but we'll add an additional data format that will make the accessing of time tag records over multiple frames much easier. So take this as a teaser. Apart from this, about traditional FLIM, so because what I presented, of course, it's exciting new advances, but still it's normal confocal FLIM. But we are thinking about ways to try to do confocal FLIM with more resolution and more contrast. And we try to, and this we achieved by implementing the image scanning microscopy paradigm together with FLIM. Of course, this is pioneered by Seppert, Enterline, Zeiss, because they have the ERI detector, and also Giuseppe Viccidomini in Genoa, that they have published a lot about the SPAD arrays. What is the unique point in Luminosa is that for the first time, and I can be confident to say that for the first time, you have a very nice, robust integration of a SPAD array in a commercial microscope together with a lifetime. Of course, also Nikon has the N-Spark, but they don't have any lifetime information. So with image scanning microscopy, you don't have to change your dyes and so on, and you get a little bit further, a bit of increase in resolution, but also a huge increase in contrast. And here you can see these results. For example, here you can see the confocal area, and here you see what one can get when implements the image scanning microscopy with species separation, including the time information. So we have more resolution, higher contrast, better sectioning, and actually you get even improved lifetime-based marker separation just because you reject the autofocus light. This is not only done with it's not something abstract that you can do this, let's say, with an external software, and we cared only about the acquisition. This is by now available in the software. So you can see here, if you apply the ISM, sorry for this, the ISM plus the deconvolution here on the left side, you can see what you will get with the confocal setup, and here you get here you see what you get with the convolution plus the contrast enhancement. So the difference is obvious. And of course, you get this information with the SPAD array, but still you get the lifetime information. Here you see the phasors, and also here you see the species separation. We plan to build more on that. So in the next, in the future, there will be even more in this direction. We try to, as a, not as a teaser, but as a, let's say, a highlighting example, we try to see how many fluorophores we could multiplex with ISM. So here we used impulsed interleaved excitation, a 485 laser, a 532 laser, and a 640 laser. And in each channel, in each spectral channel, we had 2 dyes with different lifetimes. And we tried to see if we could separate them. Right now, we display only 5 of them because the 6 is not shown here because it will be confusing to the eyes. But in reality, you can see here is the confocal separation with based on the lifetime information. Here, what you see when you include sectioning and deconvolution, and here you see what you get only with the intensities. So if you want to see it in a bigger field, you can see it here. And of course, ISM, it's only, let's say, 30% high resolution. But the big advantage is that you don't have to change your protocols, you don't have to change your dyes, and you have this big contrast enhancement. So by using the SPAD array in Luminosa, you can really think that we can say now that we push the boundaries of confocal FLIM, because you get FLIM with unprecedented contrast and unprecedented resolution for confocal system. Of course, once you have the SPAD array there, one can think about what else can be done with the SPAD arrays in terms of FCS, single molecule FRET, and single molecule localization microscopy, but this may be something for next year. And if I have a few minutes, I will just talk about how confocal, how we push again the boundaries of confocal FLIM by combining it with single molecule localization approaches for super resolution. So I have to say that this is, it requires a unique combination of speed, precision in lifetime determination, and the sensitivity. So we know that parts of the measurements that were presented here, people try to perform with other systems, and they didn't get the same quality of data. So I think this speaks for the unique combination of Luminosa. So first, we could see that even in a confocal system, we could perform FLIM storm, where we can determine the lifetime. It would get a localization precision of 10 nanometers. Maybe this is more interesting, and this is definitely something that we will expect more. So this is DNA paint, but DNA paint with a twist, because if you try to immobilize your cells on a gold surface, then there is an effect which is called metal-induced energy transfer. And I think, to making a longer story short, at the end you get a calibration curve where the lifetime of your dye can be directly translated to height. So you have a height lifetime dependency, and where you have a localization precision, in this case around 20 nanometers, we could do 3D SMLM based on the lifetime information, where we could differentiate differences. We could see difference of around 10 nanometers in Z for DNA paint. And at the end, another example that we used even FRED together with single molecule localization approaches in a confocal setup, where we could achieve, in this case, I think the resolution was a little bit worse, around 30 nanometer, 35 nanometer, because this has to do a little bit with the photophysics of the dye and so on. But definitely, Luminosa has the potential to do confocal single molecule localization microscopy, and the advantage there is that you get rid of background that you have in the wide field. But of course, one can say, yeah, but if I do single molecule localization microscopy in my confocal, I'm much slower. And this is true, but this for the case of Luminosa can be circumvented in a way, because one can set a very long tiling and stitching measurement for single molecule localization microscopy, and then leave the room and go home. So the measurements that you have seen here are mostly overnight measurements, where you could say you can set the microscope measure for 12 hours, and then you don't have to worry so much about the speed. I mean, and with this, I would like to wrap things up. I try to make it short and effective, and I'm happy to take any questions. Thank you for your attention.